In this figure, two concepts relating to DNA are presented. The first is called antiparallel, and the second is called complementary base pairing. Let's start with antiparallel. If you kind of break down or dissect the term, anti means opposite, parallel means two things that are going in the same orientation or in the same plane. So the DNA molecule is said to be parallel. In other words, the two strands are lined up side by side. So this is the parallel nature of DNA. Where does the term anti or opposite come into play? It has to do with the orientation of the DNA. Remember, the nitrogen containing base is associated with a sugar, and this molecule or this particular figure is a little bit too simplistic in my view because it doesn't really explain why I have a T and then I have a 5 prime, because we know the 5 prime refers to the sugar, not to the nitrogen containing base. So the idea is that we can simplify the structure of DNA and just say DNA, a polynucleotide strand, has a 5 prime end. That is the 5 prime carbon of the sugar molecule, deoxyribose, in this particular position. Now remember, off of the 5 prime carbon is a phosphate group. That's why this end is sometimes referred to as the 5 prime phosphate. Then we're going to go sugar, base, phosphate, to base, sugar, to phosphate, and so on. In other words, we're going to be moving along the backbone of the DNA molecule, which are alternating sugar and phosphate groups, and then extending into the center, we have the nitrogen-containing bases, thymine, guanine, adenine, and thymine again, and then cytosine here. So the arrow going this direction is representing the orientation of 5 prime to 3 prime. Now, the 3 prime refers to the 3 prime carbon in the deoxyribose molecule in this position. And you'll remember the chemical group located on the 3 prime end is not a phosphate group, it's a hydroxyl group or an OH group. And so if we look at the other strand, we can see it runs backwards. That instead of having 5 prime to 3 prime here, it's opposite or anti. We have 5 prime phosphate here going in this direction to the 3 prime hydroxyl here. So that's the explanation of anti-parallel. The second concept, complementary base pairing, is demonstrated by complementary nucleotides on the two polynucleotide strands. This is the first strand, this is the second strand. So remember, if there's a T in one position, the complementary base would be an A on the other strand. If there's a G, then the complementary base is a C. If there's an A, the complementary base is a T. And if there's a C, the complementary base is a G. This is complementary base pairing, which is the basis for Chargaff's rule in regards to base composition in DNA. In other words, the ratio of adenine and thymine in any segment of double-stranded DNA should be approximately 1, because the numerator and the denominator will be the same. And the ratio of guanine to cytosine will also be approximately 1. That's because the percentages of A will match the percentages of T, the percentages of G will match the percentages of C. And remember, the sum of those four percentages must add to 100%. So for example, if I told you a segment of DNA was 15% adenine, what's the percentage of cytosine? You would need to go, if there's 15% adenine, that means there's 15% thymine. They're complementary bases. 15% plus 15% is 30%. That means in the total section of DNA, there must be 70% of guanine and cytosine. Since each of those bases occurs in equal proportion, we would take the 70% divided by 2 for 35% total. And you'll see similar questions to this asked on activities as well as on the Reading Study Guide. So I hope this explanation was helpful, and please let me know if you have any questions.